It's a pleasure to be here. It's an honor to be here, and I'm very happy to be in this room, actually. Because for those of you who came late, there's a giant queue outside, and I actually had to fight my way through and convince them that I'm the next speaker. Seriously, they wouldn't let me in, so happy to be here. All right, so what I'm going to do is, like Hildegard just said, as the title implies, tell you a little bit about next-gen vectors. At least my understanding of these vectors, because I think this is a very, very subjective topic, and I'm sure if you can ask anyone here, we're all going to have a different opinion what a next-gen AAV vector really is and what it's supposed to do and be. By the way, my time is up since 1 minute and 7 seconds. That was fast. And just to kind of illustrate the pressure I felt when I got this title from the committee, this is the subtitle I gave you. I'm sure you recognize this quote. It's a very famous quote. It's been made by either one or perhaps both of these guys here. Jury's still out there. Problem is, and this is the bad news here, that both of them have died quite a while ago. And I'm not going to spoil the movie for you, but one of them, I can tell you, one of them is going to be resurrected in Avengers Endgame, and I'll leave it up to you to guess which one. On a more serious note, this is my disclosure. I'm a co-founder of this company, and I hold some patents, and I'm on some patent applications related to AAV vector technologies. Second disclosure, and it actually sort of works. David Wood has been flooding us with emails pointing out the importance of 16 to 9. I read all these emails yesterday night and I was freaking out, so this is 4.3 format, but it still works. All right, so last disclosure, like I said, I came in very late because of what's going on outside, so I missed most of Stephen's talk, so apologies in advance if there's going to be some redundancy here. It's probably only the first slide. I really run through this first slide, so this is very trivial now for you, just to reiterate the point that I think Stephen just made, that AAV, at least as a vector, is replication incompetent, which adds to its safety, of course, because it needs a helper virus for replication as a wild-type virus. And affects a lot of tissues, and I guess that's clear to you. The vector itself is not toxic, it's not pathogenic. The wild-type virus integrates in chromosome 19, recombinant vectors do not, but for a long time this has really caused a lot of excitement in the field about this site-specific integration. Like I said, vectors do not integrate into a safe harbor. The genome is fairly simple, which makes it easy for us to engineer the virus. There's only 3 genes. There's a rep gene encoding the replication genes, cap for capsid, and then AP for the assembly activating protein. As the name implies, it's important for capsid assembly. And I'll actually come back to the slide. This is why I'm showing this briefly here. It infects a lot of different tissues. It also infects a lot of species, so it has a very, very broad specificity. And the reason is that there's, of course, not only one AAV variant out there, there's many out there. There's the prototype AAV2 and many, probably hundreds of thousands of AAV variants out there. So overall, this really provides us with a very comprehensive treasure box, a treasure chest that we can harness and try to improve. And that's what we've been doing, what my lab's been doing, and many, many people, friends and colleagues in the field have been doing for a long time, for 2 decades or so. Trying to evolve what we thought for the longest time would already be the next generation AAV vectors, which are non-natural synthetic viruses that we engineered, which have improved properties over natural viruses. So this is essentially the concept we've been pursuing for the past, like I said, 2 decades or so, try to go from virus evolution to a vector revolution by accelerating natural evolution. This is the second to last introductory slide that I have before I tell you a bit more about what I think is really the next generation of vectors now. One reason why we harness this treasure box which is out there in nature is because these viruses differ, of course. The capsids really determine the specificity, the efficiency. And that's data from Joe Rabinowitz's lab from about a decade ago, which I think really nicely illustrate the fact that if you swap the capsid and keep the transgene persistent, you can easily retarget the virus from the lung to the liver to the muscle to the entire mouse in the case of AAV9, including the CNS. So like I said, next generation really, I mean, we are there already, we can retarget the virus, but what's the next generation? Like I said, this is subjective, so I think this is really in the eye of the stakeholder. Whoever you ask is going to have a different opinion. What I do think most of us will probably agree on is that the fact that as scientists, personally me, what interests me most is really the specificity I just mentioned, because you don't want a virus in gene therapy that targets 20 different tissues. You want a virus that is specific for one tissue, ideally for one cell type. So I think that's a big challenge still in the field to make these capsids, the viral capsids, as specific as possible. This has not been achieved, at least not in all cases. If you would ask a patient, I would guess for a patient next to specificity, I'm not sure if a patient really cares too much about specificity, but as a patient, I think people want to have a vector that can be controlled after it's been delivered. And I think this is an area which we haven't tackled sufficiently as a field yet. So because delivering is relatively easy, you can target tissues with more or less specificity, but once the vector is inside the tissue, there's no way currently to control the virus. We cannot turn it on and off, we cannot increase the expression, decrease that. We're basically completely losing control of the vector once it has been delivered. And again, I think this is really an important area that we have to tackle as a field when we talk about the real next generation vectors. if you ask the public, perhaps they would care a lot about the efficiency. They want to see this thing work, of course, and be specific control, but really efficiency, I think, is very obvious, a feature that a virus should have, a vector should have. And last but clearly not least, and I think Stephen also mentioned, is at the end, manufacturing. We should not forget about the fact that it's nice to have vectors that work in cell culture and animals, but eventually they need to be producible, they need to be manufacturable in a scale that allows application in a large patient cohort, ideally all over the world, so you have to be able to ship this virus also and store it and stockpile it and so on. There's a lot of problems here, challenges that have not been resolved yet. Above all, of course, vector safety is very, very important. I think we would all agree, all stakeholders agree on this. And last but clearly not least, and I'm already looking at Anne, so I'm not going to go much further into this because that's the next talk. Really, these viruses, vectors, of course, need to be immune evasive. We need to deal with the fact that a lot of us are already immune. We have pre-existing immunity to AAVs, or at least after a single vector administration, you are going to be immune. So re-administration is still a largely unresolved problem also. And of course, all of these go hand in hand. I highlighted here the specificity versus manufacturing. I think that's a sort of philosophical debate. A lot of companies are currently working on and have already achieved manufacturing processes that allow upscaling of virus production to 10 to the 18s or even higher titers. You could argue that, or I would argue this is not, at least not the exclusive solution to what I just said, the challenge that we need to treat lots of patients, just cranking up vector production, because eventually you're going to flood patients with viruses that are not optimal, capsids, vectors, that are not as specific as they should be probably. And I think it's not a solution to just overload patients with viruses, hoping that 1% or so is going to go to the target and the remaining 99% are going to be elsewhere. So I think we as scientists, again, have the responsibility to make these things as specific and as efficient as possible, which will eventually lower the burden on vector manufacturing. So I think these two have to go hand in hand: biology, understanding of biology, improvement of biology, and manufacturing. And of course, all of this has to comply with FDA, PEI, EMA, whatever the names are, or regulatory authorities. And communication is very important. Obviously, this may sound trivial, but I think it's important that we as scientists don't lose touch. We need to stay in contact with patients and the public and all the other stakeholders. And actually, I was not going to say it, I will say it. This is sort of an inside joke here, which only a few Germans in the audience might recognize. There's a particular reason why I put these scientists up here. This is scientists from Heidelberg University, where I come from. And this is our local Theranos story. So I'm sure a lot of you know what's going on with Theranos. So I'm not going to go into any further details, but they failed dramatically to communicate with the public and the other stakeholders. They went public, made vast overstatements which are not true, and this is really a disaster currently, a public relations disaster in Heidelberg. This is all over the German news, so I'm looking at one particular German, but I guess you don't know the story, but it's... All right. So I think really what helps us reach some of these goals here, what really has accelerated the field and led us to the next generation of AAV vectors in the past few years is the advent of next generation sequencing technologies. We heard a couple talks this morning, or I heard a couple talks probably also, where people are applying this technology. There's going to be more at this meeting and posters and talks. And I'll just summarize, I'll give you an idea of what technologies are out there and how we can use them to rapidly accelerate the screening and simplify the screening and screen much more stringently and more straightforward than we could in the past. Like I said, only a few examples here to give you an idea of this. One possibility, we typically start with libraries and in a range of a few million different capsids, and then we inject them into animals, mouse, NHPs, what have you. And here's an example where we shuffle capsids, so we create chimeric sequences over the entire sequence length of 2.2 kilobases. Inject this into, let's say, a mouse and take out a tissue of interest and then sequence whatever went into this tissue. And the problem that we've been facing for the longest time is that sequencing technologies were not advanced enough to allow us to sequence the entire capsid gene. We could obviously do Sanger sequencing, but that is very, very, very low throughput because you have to clone the sequence and sequence individually and then reassemble the small sequences. So by low throughput, I mean 20, 40, 50, 100 or so sequences. And that's, of course, not representative of a library of millions of particles. But just to throw out the keyword here, there's now newer technologies which really are being applied very intensively in the field. There's some more Germans coming in here. Hello. They would recognize these scientists because they're also from Heidelberg. So one of the technologies I just want to mention here, I have no stocks in the company, I'm just going to say it is smart sequencing technology, PacBio sequencing, Oxford Nanopore sequencing. There's a couple of these technologies. And what they have in common is really they are next generation sequencing technologies. They now allow us to, first of all, sequence the entire capsid gene. We no longer have to reassemble the capsid gene, so it makes it a lot easier for us, and they're much higher throughput. They're nowhere near Illumina sequencing. We're not talking about hundreds of millions of sequences, but we're tens of thousands of sequences. So a couple orders of magnitude above what we could do with Sanger sequencing. So this really is a major, major advance in the field. And again, I think a lot of us in the field have started to explore these new technologies to hopefully get better vectors out. I just mentioned Illumina sequencing. Illumina sequencing, I'll just click ahead, is a very convenient technology if you make libraries which are based on peptide display, where you insert small oligonucleotides encoding short peptides, 7 or 9 amino acids long, and the rest of the capsid gene is always identical to wild type gene. So you can essentially sequence the oligonucleotide and then just track library evolution and pick out your favorite candidate. So this is relatively easy, and it's a very high throughput, and this has been used in the field for, I don't know, the past 5 or so years now. We use this also in our lab extensively. Last but clearly not least, I'm going to show you a few data on the third strategy here. There's barcoding, AAV barcoding. Again, those of you who were in the AAV Vector session this morning, you heard some talks about barcoding. There's many more coming up in the next 2.5 or so days, I'm sure, because it's very powerful. What you do here is you take a smaller collection of capsids. Again, there's many flavors, but the way we do it here, you take a small collection, 100 or 150 capsids or so, which you have pre-selected, but you want to stratify this small sub-library. What you do is you stick a small DNA barcode into the vector genome, which you're going to package, ideally in the 3' UTR, so it's part of the DNA and the RNA, the mRNA, which is made from the capsid. So you can track on both levels. And then, again, you make a library, you barcode all of these capsids, inject it into an animal, and take out your favorite on-target and many off-target tissues, and then do Illumina sequencing. So you essentially sequence only the barcode, which is, depending on how you do it, 8, 12, 50 nucleotides. We use 15 in our lab. So this is, of course, fully compatible with Illumina sequencing, next-gen MiSeq or so sequencing. So you can sequence very deep, and you can track these particles, like I said, on DNA and RNA level in all sorts of tissues. You can multiplex this to many levels. So it's very, very powerful and gives you a very comprehensive picture. And I'll show you a few examples of this. There's some more animation here, which I'm going to skip. This is what I just said. So essentially, you take a collection of capsids and you barcode these. And what you do is you pool all of these to inject a single or a few animals with a whole collection of barcoded capsid variants. And then they're going to distribute. They're going to go to different tissues depending on their specificity. So again, I'll just click ahead. Now what you do is you extract the tissues, you extract the barcodes, DNA and RNA, and then you sequence on both levels just by next-gen sequencing. Then you get this comprehensive picture. And this work in our lab has been done by Jonas Weinmann, who's position number 100 something in the queue outside, so unfortunately he's not here. It's a pity. Perhaps he's happy he's not seeing this picture, I don't know. But he told me actually I should not use this picture, so I'm not making fun of him. He said I shouldn't. So, but again, very briefly what he did is and I'll just really click through this here, he made a variety of libraries, and just 3 examples here. You see the numbers of capsids are between 80 and 160 or so. And the reason it's relatively low throughput is because we have to produce each of these individually. We cannot produce them in bulk format. We package one barcode into one capsid, so it's a single vector preparation per barcode per capsid. So there's a lot of work in here. And then we have wild-type serotypes, peptide mutants, chimeric capsids, and increasing numbers of published benchmarks, because we're really interested to see how published capsids compare to each other once we run them side by side in the same animal using barcoding technology. And then what we do is we inject mice, 10 days or so later extract different tissues of interest, 20 or so different tissues, and a lot of different cell types from the liver, the blood, what have you. And again, I'm going to have to cut this very, very short because it's not supposed to be a data talk anyway, so I'll just show you one example of one capsid which we would consider a next generation capsid, which I think we would have never found without this barcoding technology, because it was hidden in our libraries that we had in the lab, but this barcoding technology actually revealed this capsid. And this is highlighted here. So this is a heat map depicting all the expression levels and specificity in different tissues, different capsids. The one that I highlighted here, I want to show 2 data slides on this. This is a very muscle-specific capsid. We weren't looking for a muscle capsid. We just screened all the tissues, and we saw this one capsid popping up, really sticking out. What I'm showing here is this is luciferase data. This is wild-type AAV9, and luciferase is a muscle-specific promoter. So you see the muscles showing up here. This is, I'm not sure if you can read this, but skeletal muscle is hit hard and diaphragm to some extent. And this is our new capsid now, which we found by barcoding next-gen sequencing technology. This is, of course, same camera setting, same dose, and you can tell really this thing, the mice, they glow. And again, if you take out the tissues, you see a dramatic improvement in the expression in skeletal muscle, in the heart, and the diaphragm. And at the same time, this capsid coincidentally is also strongly detargeted from the liver, so it's really as good as it gets. And like I said, this is this is a lucky finding here, so I'm not going to claim we rationally designed this capsid, we did not, but we applied this next-gen sequencing technology and barcoding technology, and we pulled this out of a library. I'm of course very happy to have this capsid, and just to illustrate again the efficiency. This is GFP expression now. This is again wild-type AAV9. You see wild-type AAV9 expresses a bit in the liver and the heart and a little bit in the muscle also. And this is our capsid in comparison. Again, same dose, of course, same camera settings, everything. Oops, sorry. No. Oh, it doesn't go back. What you're supposed to see on the previous slide, I was too fast, is that we essentially set the camera to the liver signal in the AAV9 mouse. So that's why, not that's why, but that was used our internal standard essentially. And then you saw the increase in the muscle, I guess, at the bottom. Too late. So I'm coming back obviously to the slide that I started with, and I want to end on making a few points, or again, just giving you a couple examples where I think beyond next generation sequencing, what else we can do to make better, more specific and safer vectors. And one thing again I cannot stress enough, I think, is that personally I think we really need to focus more on biology. And there are going to be some talks, I saw the abstracts of course, there are going to be talks where people go into the cell essentially, because that's what I was going to highlight in a minute. Really, we have to understand how the virus works. What I'm highlighting here is two papers that we published, and a lot of people work on this. Luke Vandenberger's lab and other Jürgen Akai's lab on the AAP protein, the assembly activating protein, which was found only a decade or so ago, which we've missed. The entire field has missed this protein for about 30 years. And I think it really nicely illustrates there's a lot to learn about AAV biology still, and we need to understand this biology because that's going to help us eventually to make better vectors, rationally design better vectors, and then again lower the burden on manufacturing and improve the safety for the patient, of course. So as I just said, the cell itself, I think this is really still a black box, how the virus interacts with the cell. And we heard a bit this morning, we're going to hear more about this, how it binds to cells, how it traffics to the endosomes, to the cytoplasm, how it gets into the nucleus, how it's uncoded, a lot of open questions. And once again, we need to understand this really to make better vectors. What I think is really exciting as a new technology or a new goal is to further increase the specificity beyond targeting organs, but really targeting specific cell types within the organ. We and others have done this, for instance, in the eye or the brain. You don't want to target the brain as a whole, but of course you would need to hit neurons or astrocytes. So, but also in other organs like the liver, a lot of people target the liver as a whole organ, but again, the liver is a complex organ. I'll show you one data slide on this, where I think it's really important to be able to target hepatocytes or stellate cells, but not the entire mass of cells in an organ. And this is actually one example here where we pulled out a capsid, which is very specific for stellate and Kupffer cells, but it's detargeted from hepatocytes and also from the L-sex endothelial cells. Which struck me in the AAV session that I attended a few hours ago, what was shown here, I think selection on the RNA level is extremely important. And it struck me because there were 3 out of 6 talks or so where they made the exact same point, which is good, of course, just shows we are thinking alike. And there's been a number of technologies, I was trying to remember, I think it's tracer and trade and iTransduce, all fancy names. What they all have in common is that people start selecting on the RNA, not longer the DNA level, as we've been doing as a field for, again, a decade or so. Because screening on the DNA level gives us a lot of false positives, viruses that attach to cells are stuck in the cytoplasm, but they don't reach the nucleus, they don't make mRNA, and that's what you want, of course, from a next-gen vector. A vector that really expresses strongly, and it doesn't only interact with the cell in some manner. So I think this is a very important thing to sequence on the RNA level. One technology to do so is single-cell RNA-seq, and I'll show you if there's still time. By now I'm 18 minutes over. If there's still time, I'm going to show you 1 or 2 data slides on single-cell RNA-seq, which I think is a super powerful technology and very cool. And it gives a lot of new opportunities for screening and capsid evolution. Combinatorial strategies, what I mean is here we have to make the best out of the very limited space which we have. And again, there's been some indications in talks where people really work on promoters and poly-A signals, try to minimize whatever they can to squeeze as much as we can into a capsid. And the example I'm showing here is we multiplex, so we combine RNAi and CRISPR in the same capsid because it fits actually, and we use this to target different infectious pathogens at the same time using RNA and DNA nucleases. Fine-tuning, this is the point that I made initially where I think not enough work has been done yet but needs to be done for the next generation of AAV vectors. Again, getting control of the vector once it has been delivered to patients, how do we turn this on and off, how do we eliminate the vector? If anything goes wrong, we need safety switches, kill switches to get control and keep control of the vector. And again, a few examples, keywords here, I'll show you one slide. I also want to mention AAV is not the only parvovirus out there. There's other viruses like Boca viruses. And again, we just published on this. Other people also work on this. You can cross-package an AAV vector genome and capsids from different parvovirus. And again, it gives you a lot of options to optimize specificity, efficiency, and so on. And I think we should not forget there's really novel applications out there. It's not only about cDNA delivery, RNAi, or CRISPR. So one application I find personally very interesting is reprogramming transdifferentiation. And again, I think not much work has been done on this, but I think this is, again, a very open, a very new, and a very wide field. Like I said, I want to conclude, if I may, on, 5 minutes. I thought I have like half a minute or so. Okay, I want to conclude on giving you a few ideas what I just said, basically what can be done. Okay, the screen here is flickering really bad. About the single-cell RNA screening that I mentioned, which not too many people have done, what I think we need to do as a field, focus on individual cell types. So what we've done here is what we've been doing for the longest time, make a shuffled library, inject it into a mouse, take the liver, and then see what gets in. But again, like I said, the liver is a complex organ. There's these cell types and actually more cell types in the liver. So what we did here is we isolated, purified these cell types by FACS or MAX, and then did PCR rescues in these individual cell types. Then make a library and go back in and do this multiple times in a row. And indeed the battery died or something. No, here we go. This is very slow now. Jesus. Okay, just skipped it. But you see what's happening here, right? Yeah. Oh, God. Okay. What I meant to say is that by doing this, we can extract or isolate particles, as I was briefly showing you on the previous slide, that have a higher specificity for an individual cell type. The problem with this FACS-MAX-based approach is sorting-based, so it's never pure enough, and there's, of course, not markers for all cell types of interest, so there's some disadvantages here. And this is why, as I said, we've begun a few months ago to work on single-cell RNA-seq, and this works, in my opinion, really tremendously well. This is very, very exciting. So we can isolate the cells and identify the cell types from the liver based on the transcriptome. You see what we're not supposed to see, some dead cells, some red blood cells. It's not perfect. We have these in there, but we can identify Kupffer cells and stellate cells, hepatocytes, and the endothelial cells, T cells, and many more fractions here in the liver. Then what we do is we combine this, like I said, with barcoding technology, so we can identify these cell populations and then use our barcoding strategy and assign the barcodes to the transcriptome and thereby to the individual cell type. And what we can do then, of course, we can rank the particles, we can see which cell types they preferentially transduce. This is shown by cell type here, and see which particles go into a particular cell type. Or the other way around, we look at the virus and see which cell types are transduced. And what you can, for instance, appreciate is that AAV8 in our DJ capsid, a shuffled capsid, have a very strong preference for hepatocytes. Other capsids like AAV6, they hardly transduce hepatocytes, but rather they go to Kupffer cells or stellate cells. Again, this is very, very high level here, just to give you an idea, and I think this is really a technology that we, of course, and probably also many colleagues and friends in the field, will apply in the future, because it really gives us a completely new level of resolution and of depth also. Just one point I want to make here. I'm not sure if there's much talk about this meeting here. I saw there's a talk on antibody retargeting coming up. We have to keep in mind, AAVs, of course, they have a shell, they have a genome inside, but there's more than we can do with a particle. Rather than engineer what we have, we can stick moieties on top of the capsid. Peptides, nanobodies, antibodies. We use nanobodies here, so basically the heavy chain, the variable domain of the heavy chain. This is collaborative work, and again, only just to give you an idea, we can stick the nanobodies. Either we can make bispecific nanobodies which bind to the AAV capsid and a cellular receptor, or incorporate the nanobody into the capsid really, and then this is, I guess, the smartest and the nicest way to do so. And again, just you see from the data, this is very, very effective. We can retarget the viruses very efficiently and very specifically to cell types expressing the receptor which we're targeting by the nanobody. And like you see, the number is 60 or 80% or so. Last point I want to make here is CRISPR, of course, or the combination of AAV and CRISPR is very hot. A lot of groups are working on this, including ours. Problem with CRISPR, you heard this probably also if you attended the gene editing workshop, there was a lot of talk about the size issue with CRISPR. Of course, the Streptococcus pyogenes Cas9 is too large for an AAV vector, it's borderline. So what we did is we split the Cas9 into two double-stranded vectors, and we use Intein technology. I cannot go into details, but it's essentially a splicing mechanism on the protein level, so we can have all the benefits of a double-stranded AAV vector, and I think this is an example of smarter vector design. And we get very robust and very strong and rapid Cas9 expression and CRISPR activity then. And we can combine this with what I mentioned, which is the last point I want to make here, the safety switches, kill switches. So essentially guide RNAs which target the Cas9 itself, and then the vector inactivates itself. And this is shown on this slide here. So you have an AAV, it goes into the cell, and eventually you want to turn this off, and this is where these self-inactivating switches or kill switches come in. And this is just showing the principle, so it actually is fairly simple. You make a vector and you include a guide RNA against the vector itself, and the transgene can be any transgene or a Cas9 in the case of CRISPR, and then as soon as you add the Cas9 as a protein, as an mRNA, as another vector, it activates the guide RNA and the vector cleaves itself and is inactivated. In the absence of Cas9, of course, the vector just continues to fire for whatever period. This just shows it works over 2 orders of magnitude. It's very, very potent, this technology. And this is really on the last slide, just showing this is too busy, this slide here, but the idea I want to give you a take-home message is a very, very versatile system. Like I said, it works with any transgene, be it CRISPR or anything else. If you insert this kill switches and activate them with Cas9, you can turn the vector off at will at any given time point. And this is the advertisement. I only want to say that they're actually here now, right? This is the people that were running in front of me a few minutes ago. 2 colleagues and friends also. This is a group leader, Dominik Neopek in Heidelberg, and his PhD student. And what they work on, they use microRNAs to fine-tune AAV vector activity, especially in the context of CRISPR. Yeah, it's just illustrating the situation here. You want to have a vector, like I said, which is very specific, active in one cell type, not active in the other cell type. You can use microRNAs to turn it on and off. There's many strategies out there. And what they have done, I'm not going to tell more about this because they have talks coming up, but they've essentially combined this with CRISPR, an anti-CRISPR protein, put the anti-CRISPR protein under control of these microRNAs so you can turn this on and off in certain cell types. This is strategy 1, and this is presented by Mareike, the PhD student, on Wednesday morning at 9:15, so I really recommend to go to this talk and see the details. And what Dominik has done also with Mareike together is to put this under light control, under control of blue light, and again you can turn on and off the inhibitor of Cas9 and thereby control vector activity. And this again comes back to the point I said, once the vector is in, we need control, and I think this is 2 beautiful examples of how this could possibly be achieved. And also Dominik's going to have a talk at 9:30 right after Mareike's talk, but also on Wednesday morning. Having said this is the slide I showed before, and I know I was going very fast and time was very limited, but I hope I could give you an idea of some of the technology strategies which are out there. There's many, many more. This is not exhaustive what I told you, of course not. And I apologize to all colleagues whose ideas I have not mentioned here. I couldn't. And like I said before, I'm going to hand over to Anna now to talk about this particular aspect. Thank you.
Thank you very much, Dirk, for this wonderful talk. This was exactly what we hoped for. So I think there are a lot of questions for Dirk.
With all the screening process used with barcoding and sequencing, it's all done in the mouse platform. Is there any confidence that it will actually translate whatever you select to actually the same timing?
I think that's the killer question that was also discussed in the AV session. It's a very, very good and a very important question. I have no firm answer. I can tell you we did evolve some of these capsids in mouse, and we took the lead candidates into monkeys. This is going to be a very vague answer because this is done in collaboration with the companies. I cannot really disclose the details here, but I will say, yeah, in some cases it does hold true. In other cases they behave very differently between a mouse and a monkey, which I guess is not surprising at all. So very good question, but I have no good answer.
Is there any new generations of the screening process that may make that more better translation?
As I was trying to imply, I mean, I think what we did in this mouse work that I just mentioned, again, we screened on the DNA level, which I think is not good. We should have screened on the RNA level. And I think the best we can do probably at this point is screen on the RNA level directly in non-human primates, cell type specific. Just, sure. Okay.
So I don't see any further questions. So thanks again.
Thank you.